Okay, so today we continue our lecture on chemical sensations, and yesterday we have considered taste, so today we are ready to transfer to the olfaction. So we should be ready to talk how do we smell different things and so on. But before we get into that, I want a little bit return to the yesterday's lecture because we had pretty interesting question regarding the batteries. Why do we taste batteries? And I guess it's pretty interesting. And yesterday I had some hypotheses regarding the electrolysis. That electrolysis gives as a result some protons and maybe some some OH minus groups, and that can be tasted. Well, at least protons, they should taste sour. That's good. Now I have found at least some resources on the internet. So here is a video I have added to our presentation in the addendum. I don't think we will go through all of that video, but basically the explanation aligns with my yesterday's answer regarding the electrolysis. So you may check this video details after the lecture.
Or to that sage advice, in addition to the slight, it was actually the taste of the electrical current from the charged battery. When you touch the little knobs or leads of a 9-volt battery on your skin, you don't feel a shock because your skin doesn't conduct hydrogen gas, leaving hydroxide ions behind. And current leaving your saliva through the other lead creates oxygen gas, leaving hydrogen ions behind. These hydrogen ions are what sour taste receptors in your taste.
So that's basically the main body of the explanation, that we have hydrogen ions created at one of the electrodes, and these hydrogen ions, they can be tasted. Now you might guess I can't stay aside and not create for you some sort of tricky question that we may try to consider right now. First of all, let's think what is happening on different electrodes. So let's remind ourselves what is happening during the electrolysis. So we have considered some sort of chemical part, and you probably might guess what should be going on the plus terminal of the battery and on the minus terminal of the battery. Who will help me to fill this in? Mm-hmm. Well, basically, if we have two terminals of the battery, so there is some influx of electrons or efflux, depending on what terminal we are talking about. If that's a minus terminal, so the electrons are getting into the solution. So let's just consider some pure water. It's not really practical case, but let it be. So suppose just pure water, and we are dealing with this sort of situation on two different terminals of the battery. So I can remind you that water may undergo this sort of process. So we have H plus OH minus Now what is the half reaction at each of the electrodes? So probably the easier is the reaction at the anode, so the or the cathode. No, probably the cathode, the minus sign. So let's consider this one. So if we have some molecule of water that has undergone this sort of reaction, so now we have H plus, we have OH minus. This minus terminal tells us that we may want to add some electrons, so we are pushing electrons into the solution. Who of these two guys will be happy to accept an electron? H, yes, absolutely correct. So H plus has no electron. He will be happy to get some electron for himself. Now we have neutral H. That's pretty radical, pretty radical thing. So probably he will find another one H and combine into H2, creating a hydrogen molecule. So what we expect here is something like that. So we have I guess two molecules of water, so we have 2H2O that gets 2 electrons, and as a result we get some hydrogen gas evolving, so it just flows away, and we have some OH minus. So pretty reasonable reaction. Now what happens at the plus terminal of the battery? At the plus terminal of the battery we are hijacking some electrons. Which one of these two guys is able to give away an electron? OH minus, yes, absolutely correct. So when water undergoes this sort of reaction, OH minus will give away its electron. We will get this sort of OH radical. There is some sort of a bit complicated mechanism. They will combine into a peroxide. That peroxide can, well, depending on the conditions, either go further with some sort of electrolysis or evolve some oxygen atom. So all in all, we will get something like this. So 2H2O. I guess nothing is needed. We are abstracting some electrons, so we will get O2 evolving. We will get 4 protons, and we will get 4 electrons that will just go away through the electrode. So here we have some protons created. Here we have some OH minuses created. So that's what we have. Now comes the tricky question for you. Whenever we have some hypothesis, it's pretty reasonable to find some way to test it or to falsify that hypothesis. Otherwise, that's not real science. So I don't want to get into the pure details, because that's probably more like of a philosophy of science. But maybe you have heard like something like Popper criterion or something like that. So the idea is, whenever I make some claim, there should be at least some chances that someone can test that and somehow show that this claim is not correct. So why not? So where am I leading you to? To the idea. So let's devise some experiment, some very simple experiment uh, that will at least somehow test this hypothesis. So we don't expect some sort of perfect testing like giving the final answer, but at least something that we can quickly test, and if that fails, we will know that this explanation is not correct. Can you come up with this sort of experiment? Please share any ideas. Number of batteries. Well, okay. Suppose we will know that this sort of flicking, so like in this image, gives you some sour sensation. But we are still unsure regarding the mechanism. Maybe you are sensating, I don't know, some add electrons to the water and check acidity. Well, interesting idea. I'm not sure how to do that. I don't think I have some sort of that pepper thing to just add them to the water. uh, but but that's pretty close. Well, at least to something I came up with. So what can be some sort of alternative hypothesis? Yeah, let's first before going in, let's come up with some alternative hypothesis. So what can be the reason for some sort of strange sensation from the batteries? And then we may devise some experiment. I know we are some sort of stepping aside from our lecture, but I guess it's pretty instructive because that's how you will tackle any problems in the real world. So so can it be that we are sensing, I don't know, some metal from the context of the battery? Maybe. Well, sounds interesting. Uh, but what if just tasting the battery? So the hypothesis number one, yeah. So we may be tasting some metal. Uh, a good idea. So let's taste something different with electric current, and maybe we will be able to feel something. Okay. What about other alternative mechanisms? Suppose that electric current just activates some neurons. So we know that neurons, they generate some action potentials and so on. So maybe this electric current is able, I don't know, to depolarize something or somehow cause the neuron to fire that action potential. So it has nothing to do to the electrolysis. So we are just some sort of electrocuting our neurons. Well, in fact, we a little bit we are, but can you come up with any interesting idea regarding this sort of thing? So can we distinguish these two things? Check brain activation. And what will that show us? So okay, so some parts of brain are activated, but probably we know that already. So we are tasting something, we feel some taste, so probably some appropriate brain areas are activated, otherwise we won't be able to taste anything. How do we distinguish the electrolysis hypothesis from just some sort of activating neurons with electricity? If you look at these formulae, so let me go to this slide, it's just better view. Good, sounds interesting. So why don't we block our proton receptors, and then if we are just activating our neurons with some sort of electricity, that will show us that we have will distinguish between these two cases. Yeah, that's a good idea. Anyone else has some more ideas? Nope. Good. Here's the thing I came up with, and probably some of you probably think the same way, but just don't want to share your ideas. Please look at these two half reactions. So one is going on near the plus terminal, the other is going on near the minus terminal. So we know that the taste is sour. Sour taste, that's basically taste of the protons. Let me choose another color. So that's taste of the protons. Here are the protons, and here are none. So that hints us that probably different leads of the battery, they should taste differently. So if you take some sort of battery, some battery like this one, so here is a battery just 1.5 volt. I'm not sure if everyone will be able to taste that. Well, I can, some other people not so much, but if I complete the circle by touching only one terminal with the tongue and the other terminal with, let's say, a finger, just to complete the circle. So if I do that this way and I touch it, I can feel some sourness because that's a plus terminal and there are some protons that can be tasted. If I do that the other way around, so I'm touching with my finger the plus terminal and I'm tasting the minus terminal, there are no protons. OHs are formed there, and that's it. So basically I don't taste that sourness if I taste it this way. So that may be one of the ways to quick test the hypothesis that it has really to do something with the electrolysis. And here are some videos for you. I'll just quickly turn it on. So here is a video of performing electrolysis just with a simple battery and with some indicator inside a petri dish. So I'm not showing you the whole video.
I will try again. I'm going to put my electrolysis device the same as I did before.
After the lecture, but here you see the device.
But you'll notice that in addition to the bubbles forming, as they did before, something else is going on. There are colors forming around each terminal. There's a group.
So here you see some sort of color change. So some colors are forming around each of the terminals of the battery. So here is the plus terminal, and here you see some red color that indicates some acidity. And here is the minus terminal, and here you see some change of color that indicates it's going the other way to the basic part of the pH spectrum. There's a whole long video, I guess. If you like some sort of homemade experiments, maybe you would like to check that, because all the equipment is like this, some sort of rubber bands and bolts and something like that, but that pretty works. Even the indicator was made from the red cabbage. So if you remember our lecture on the pH, I have shown you the red cabbage, and there are certain chemicals inside that may change color due to changes of the pH. So we have considered it in that lecture, so you may always return and
greenish,
check
bluish
that if you
color
wish.
forming at that negative terminal and a pinkish color forming at the positive terminal. This electrolysis device.
And maybe just one more video. I didn't find the description, the exact description for this video, so I'm not sure what are they electrolyzing and what's the indicator they are using, but once again you clearly see two spots that indicate difference in the pH. Okay. Okay, I hope this like small problem, small practical problem will have some, I don't know, some value. Good. Now we are ready to get to our lecture. Let me just check how much time do I have. I still have some. Well, or maybe you have some question regarding that problem, or comments if you wish, before we dive into the new topic. Okay, no comments, no question. Good. Then we are going into a new topic regarding the olfaction and smells. Once again, we start with a little bit of anatomy, just to position where are we in the whole organism. I guess would be reasonable if I return just for a moment to the slide we had here. So here's a little bit better view of the whole head. So here you see the nasal cavity, here you see the nasal cavity, here you see the other parts, and we will be dealing with this part. So this part we are now operating right there. Then we will move to some pathways in the brain, but that's the part. And now I'm returning here. So here you see this area enlarged. So now you can see this area enlarged. So here is the brain, here is the olfactory bulb. Yesterday we have considered some of the 3D atlases, and I hope you more or less remember the position of that olfactory bulb, or you can always return to that atlases and recheck that. The interesting thing, the lamina cribosa, or cribriform plate, here it is. That's a pretty interesting structure. So here you see the things enlarged. That's the cribriform plate, and you see these sort of holes in that cribriform plate that the olfactory receptor cells are able to use to communicate with the olfactory bulb, and this way transfer information to the brain. So that's basically the location of the olfactory epithelium. You see how this epithelium looks like. So that epithelium consists of a layer of olfactory receptor cells. What's interesting, these are neurons. It's pretty unexpected, I would guess. There are some sort of supporting cells, so here we see some supporting cells around these neurons. Probably you know that neurons, they don't like, let's say, live on their own, so they often need some sort of support. Especially neurons in the brain, they should be supported with glial cells, but that's not the topic right now. What else? And there are some basal cells. So here are some some basal cells. The odorants, they dissolve in the mucus layer. So here we have some mucus layer synthesized with the supporting cells. Here you see the cilia of the olfactory cells. So if we have some odorant inside of our nose with inhaled air, it will dissolve in the mucus layer, contact the cilia of the olfactory cells, and the axons of the olfactory cells that penetrate basically the bony cribriform plate will send some signal to the brain. What's interesting here, you see that these neurons, they are, let's say, updated. There are not much places in the human's organism where you can see some sort of neurogenesis ongoing. As far as I know, I can think about 2 such places. Maybe there is some more, but that's really rare case to find a place where some neurogenesis is ongoing in the human's organism. The one place, that's basically these olfactory epithelium. So these neurons, they can be replaced. They live for some period of time, then they are replaced. The other place has to do with our memory. That's, I guess, it's called gyrus dentata, and that's well, I'm not. I don't think we should get into that details, but it is associated with some sort of memory processes. And if you want, you can Google the gyrus dentata. So that's it. Good. So it's a pretty interesting fact that these neurons can be, in some sense, regenerated because we have some ongoing neurogenesis. Good. So regarding the olfaction, of course, it's pretty important for us because that enhances our food identification, and it may warn some of danger. So we often feel some unpleasant smell of some spoiled food, and that basically tells us it's better not to eat that. And it's interesting that humans can detect hundreds of thousands of odors. The interesting part that this encoding is performed with, let's say, population encoding. Some different neurons are detecting something, and that sort of population encoding. Oh.
Recording in progress.
Oh, I'm sorry. Something went wrong with Zoom. Okay. I hope that was just some transient issue, not something that will bother us throughout the whole lecture. So okay. Good. So olfactory epithelium I have shown you. I guess let's look how that looks like under the microscope. So here you see a sensory neuron around all that supporting cells. So here it is. Here is the upper part of the olfactory neuron. On the left, you see that it sits over a layer of neuronal stem cells which generate new olfactory neurons during our life. And on the right, you see the hair-like cilia that protrude from the tip of the individual neuron, and the receptors for odors, they are located basically on these cilia. So that's, once again, we are positioning ourselves in the whole organism, but this time on the level of individual cells. So here you see the compartment of the cell that is performing basically the olfaction. Good. Here is a more biochemical view. So what do we have here? So what is basically going on? So here you see our receptor cell. Here you see the signal will go to the olfactory bulb. I think I will mention something regarding that bulb. And here we have some receptor cilia in the mucus. So these cilia, that's what we had in here. So here they are. Okay, so what is happening? We have an odorant receptor protein. I'm not sure, I guess I have numbers somewhere in the slides, but something like 400 genes encoding different odorant receptors are in the human's genome. So there's a large pool of different genes that encode these sort of odorant receptors. They can be, let's say, pretty interesting. On one hand, they can detect, let's say, different chemicals because we have this sort of population encoding, so it's not like one receptor for only one specific chemical. On the other hand, they can be specific enough so that one receptor will detect one of the isomers of certain chemical and not detect the other isomer of that chemical. So I guess I have some example for you, so probably I will show you that as we go. So here you see this sort of receptor. You might guess that this is a G protein coupled receptor, so this is a GPCR. Once again, pretty well known to us biochemical pathway when G protein is activated, and then it activates the adenylyl cyclase. Adenylyl cyclase, as you know, produces cyclic AMP. So pretty well known second messenger in the cell. So it takes part in many different biochemical processes, I mean some signaling pathways, and this is one of them. And what's interesting here, we have certain channels that allow calcium and sodium to get in that detect cyclic AMP. So the cyclic AMP is detected by these sort of channels, and they allow the influx of the appropriate ions. These ions may additionally activate some chloride channels, and at the end of the day we get the membrane depolarization. And you probably remember from our previous lectures, if we have membrane depolarization, if we get to certain threshold, we can fire an action potential. So if that happens, the neuron will fire an action potential, and basically with that action potential transduce information that we are smelling certain chemical or one of certain chemicals that it is able to detect. So you see that olfactory receptor neurons, they use a single main transduction pathway located in their cilia, so that's what was on the previous image. Here you see the sequence of the events. So just reiterating, so we have odorant molecules dissolved in the mucus and they bind to the receptor that activates G proteins. They have some specific G proteins called G-olf, so from olfaction. So G proteins may be pretty different. They activate adenylyl cyclase. Adenylyl cyclase produces cyclic AMP. Cyclic AMP binds to cyclic nucleotide gated cation channels. These cation channels allow sodium and calcium to enter. The depolarization begins. Calcium opens calcium activated chloride channels. Due to internal high chloride, chloride flows out, further depolarizing the neuron. Threshold is reached, action potential is fired to the central neural system. So that's basically what we have just seen in this image. Of course, if we start a certain signaling pathway, we need to find a way to terminate that signaling pathway. Well, that may be just due to the odorants just diffusing away or being broken down by mucus enzymes. So we don't smell something forever because maybe that chemical just we have went away from the source of that chemical and we don't need to smell that. Rising the calcium activates some feedback pathways that reduce the cyclic nucleotide gated cation channel activity, so CNG for short. The receptors may show adaptation, so response weakens within 1 minute even if odor persists. So you probably all know that from the practice. If you come in, I don't know, in the room from the outside, you probably feel some smells of something. 1 minute passes and it looks like there is nothing interesting happening. So that's because of the adaptation. So we are pretty quickly adapting and we just stop smelling certain chemicals. Good. What's interesting, one more interesting to reiterate, that olfactory receptor neurons are true neurons. So they are capable of generating action potentials and so on. So that's maybe a pretty interesting point. So you don't have some specialized cells to perform all that olfaction and then just transfer information to neurons, like that was, for example, in taste. So in taste we had some cells that were performing detection and they were connected to neurons and then neurons performed all the information transfer. Here we have the real life neurons that do everything. Okay. Here we have some voltage recording from an olfactory cell during some sort of stimulation. So here you see some odorant, and here you see cilia with all that receptors. And you see how different potentials look like. So here is a receptor potential in the cilia, and here you see action potentials fired by the neuron itself. So odorants, they generate a slow receptor potential in the cilia, and the receptor potential propagates along the dendrite and triggers a series of action potentials within the soma of the olfactory neuron. Finally, the action potentials propagate continuously along the olfactory nerve axon. So here is an axon, and they propagate through that axon. So hopefully you remember our lecture, I guess 10, regarding all that action potentials and so on. So probably this is nothing new for you. Good. What else? So what are odorant receptors except they are GPCRs? They are basically membrane proteins with some extracellular binding sites. Well, pretty reasonable. They have some enormous diversity. So you see that rodents have more than 1,000 receptor genes, so largest known gene family. Humans have 350. Yeah, so I was a little bit incorrect when I told you 400. Yeah, so it's 350. Still, that takes 3 to 5% of our genome. That's pretty large. And they were discovered, and there is a Nobel Prize for that in 2004. So each olfactory receptor neuron expresses only one receptor gene, so the rest are silenced. That creates around 1000 distinct receptor cells in mice and probably around 350 in humans. So olfactory epithelium is divided into zones, each expressing a subset of receptors. I guess I have an image of these zones somewhere. So yeah, so here is an image that basically the map of the expression of different olfactory proteins on the olfactory epithelium of the mouse. So here is a slice of that, and these are different zones of the groups of genes expressed. So there is some sort of spatial clustering of that sort of gene expression. And within the zones, receptor types are randomly distributed. So we have some large zones. That zone takes care of some group, some subset of receptors, but within each zone you have like a mess. So whatever is happening, that's it's totally let's say messed up, so mixed up. Good. What else? There is one more system, so-called vomeronasal system. If you have ever heard a term pheromone that has to do with this sort of system, it's also called the Jacobson organ. I will show you in a while how that looks like. But this organ, if present in an animal, it uses a different receptor proteins, so much fewer in number, and they are likely specialized of detecting the pheromones. Here you see the opening of the Jacobson organ of a cat. So that's so this scary beast, that's basically a cat, and here we are just looking inside of the mouth of a cat who is probably trying to bite someone. And here you see the opening of the Jacobson organ. The organ itself, it's positioned in up there in the nasal cavity. What's interesting, it seems like the this organ is absent or vestigial in most mature people, and even when that's identifiable, it does not appear to have any functional connections to the brain or any functional proteins able to detect anything. So it doesn't really mean that we don't have any similar system with some pheromones, because we still have the major pathway through the nose, and maybe this system was just redundant and removed. We just use the other one, but still at this point we don't have any evidence that we do have any such system, so that still remains sort of controversial. So if you ever get any info regarding some pheromones in human species, that's probably a bit controversial, because we still don't have any relevant evidences that we do have one. So maybe we do. We don't have enough evidence to claim the opposite, but at this point it looks like that it's safer bet to say that we don't have one, because otherwise why didn't we find anything for so long? Okay, what else? Population coding. The mice have something like 1000 odorant receptors, humans 350, and they are in some sense broadly tuned, so each one of them is detecting some set of chemicals. The interesting part is that it may detect chemical but ignore another very similar chemical but detect some other non-similar chemical. So that may be the case. So that's pretty interesting. Uh, So we know that one receptor may respond to many odorants, and one odorant may activate many receptors. So that's what we want to get some sort of population coding that adds some stability to our system. If we have just one receptor detecting one odorant, that may be a problem. What happens if that neuron dies for any particular reason? We will be not able to detect that chemical, and what happens if that chemical is of some, I don't know, some value to us. So maybe that tells us that something really bad is happening, that, I don't know, the food is rotten and we don't need to eat that because we will just die. So that's pretty bad, and that's why the nature uses here a population coding. So when one smell activates a lot of different receptors, and different smells activate different subsets of receptors, and then the brain is able to take that information and to get some idea what is happening there. So what are we smelling? So maybe some detectors will not activate when they need it, but the other will activate, and the brain will get, aha, this pattern corresponds to that chemical. Well, there should be one or two more receptors, but probably that's it. And we will respond appropriately to the chemical. So the brain interprets the full pattern of activity across the receptor population. So not one receptor, but the receptor population. And here is an example, I guess, for that sort of broad tuning. So here we have each receptor cell that expresses a single olfactory receptor protein. So here it's coded with the color, and different cells, they are randomly scattered within the region of the epithelium. So here we have some epithelium, and here are different receptors. And here we see some sort of microelectrode recordings from 3 different cells that here's receptor 1, receptor 2, receptors 3, and here we see different smells that were detected. So some citrus, floral, and peppermint and almond. And you see that they all are somehow responding to each of these smells, more or less. So what matters is the overall pattern across all the receptors. So receptor 1, it's more like some citrus, but it detects almost anything except for the almond. Receptor 2 is able to detect citrus and floral, but is pretty hesitant to activate on peppermint or almond, while the receptor 3 does pretty the opposite. And you might guess, if we, for whatever reason, we lose one of these receptors, we will be probably still able to decode what are we dealing with. So if I remove any of them, just remove it, I don't know, something happened, that cell died for whatever reason, there is still enough information that allows me to differentiate between these sort of smells. Of course, in practice, the brain operates with many different neurons, so there's even more information so that we can get what we need from that. Good. What else do I want to show you? Let's get to the olfactory bulb and what we have here. So up to this point, we were considering this part. So that's olfactory receptor cells. These receptor cells, they send their projections through the cribriform plate. So here it is, the lamina cribosa. So here we have it. And they go up to the olfactory bulb. So here is the olfactory bulb that we have seen yesterday in that 3D views different. So here it is in the image, And here we have the interconnection between the olfactory neurons that are able to detect smells and sense some action potential, and neurons of the olfactory bulb that will take these action that will take that information from that neurons and transfer that to the brain, to other structures of the brain. The places where these interconnections between neurons, between olfactory receptor cells and neurons of the olfactory bulb is performed, they are called glomeruli. So this is one glomerulus, and here we have all these sort of connections. Let me check just a moment, maybe if I don't forget to tell you anything useful. So yes, each bulb contains something like 2000 glomeruli, so 2000 places where they do interconnect. Each glomerulus receives many receptor axons, 25,000, and they project onto 100 second order neurons. So that second order neurons, so they collect information from many receptor cells. What else? Interesting part is that axons from receptor cells expressing the same receptor gene converge exactly onto 2 glomeruli per bulb, or no more than 2 glomeruli per bulb. So that's pretty interesting because that's the only place, as I told you, that where the neurogenesis is ongoing. So if one neuron dies, we need to create another neuron here. So we need to create another neuron here. It's not so a problem to send something here, but we need some projection, we need some axon to get through the cribriform plate and to find its own glomerulus. So this is a pretty interesting thing that happens all the time. So these neurons, they are definitely knowing where do they need to go and how do they need to connect, so that connections are perfectly restored after one neuron dies and we are replacing that neuron with some new neuron in the olfactory epithelium. So that's interesting point to notice. And here are I guess some images that show you how this happens. So here you see the olfactory neurons from the epithelium. The cribriform plate was removed to show you only the neuronal part of that, and here you see that each glomerulus receives input from only from receptor cells expressing a particular receptor protein gene. So receptor cells expressing a particular gene, they are color coded, and here you see how that looks like. What's more interesting, let me show you this image. So these are neurons with a particular type of a receptor. So we are able now to perform some sort of genetic engineering and to force neurons that have a certain type of a receptor to express something else, let's say some protein that we may see under the microscope, so we can like see some color and understand what is going and to what direction it is going. So here on the left you see that neurons that express this sort of particular receptor, they are randomly scattered. So there is no visible pattern of these neurons. They are not grouped into one place, they are just scattered all over the place. But if you look on their axons in the olfactory bulb, you see that they all converge to one place. So here they all are converging, and so you get these sort of glomerulus. So this is one glomerulus and they have converged there. So it's pretty interesting mechanism, so they are able to find their way to the right glomerulus and create some connection in that glomerulus. I'm not really sure regarding the exact mechanism how that's performed, but maybe you can come up with few of that mechanisms and get some ideas. Good. So now we have somehow detected some chemical, so we know that there is a chemical and we are ready to send signal to the brain. Now where are we sending that signal to? Here is once again the slice of the brain that corresponds to the images I have shown you yesterday, so you may just find one of them if you need the name of any structure in here. While on the right you have a little bit more details. So here is our olfactory receptor cell that has detected some chemical and sends a signal to the olfactory bulb. Here we have another neuron that picks up that signal and that relays that signal to other parts of the brain. So one is the olfactory tubercle. So here it is. Here will be another connection. Then it will be relayed to the thalamus. Thalamus is known with the different nuclei. So it has many different nuclei performing whatever they perform. But medial dorsal nucleus, here it is. So we will have some projection in here. And from these medial dorsal nucleus, it will go back to the orbitofrontal cortex. So that's basically the parts of the brain where we have some processing of the olfactory information, and that information is collected with other modes of the information we get, like what we see, what we hear, so on and so forth, and create some sort of image of the surrounding environment in our mind. So axons of the olfactory tract branch. They branch. So here you see this branching. They enter many regions of the forebrain, including the olfactory cortex, and the neocortex is reached only by a pathway that synapses in the medial dorsal nucleus of the thalamus. So that's that's what we have. Good. What else should I? Let me check just for a moment so I can understand the time and the number of slides left. Well, okay, not so many are left, so that's good. Okay, so here are some ideas regarding the principles of coding. So the first one, the population coding, that's basically we went through. So no single olfactory receptor uniquely identifies an odor. So an odor is identified by a pattern of the activated receptors. Of course, we expect for each unique odor some unique pattern of that receptors activated. And the brain interprets these patterns across the population. Like a citrus odor that evokes some mixed responses in several receptors, and only a combined pattern distinguishes that from floral or peppermint. And we have already considered that receptors with these cells with the same receptor, they converge into the same glomeruli. Good. What else? The glomeruli, they form some neural map of the order that is interpreted with our brain. It's a pretty interesting moment because odors, they don't have some spatial structures that is different from anything we can see, for example, but anyway we still use some sort of maps to interpret that. So that's pretty interesting. The other two things, basically, that's the temporal or spike timing codes. I guess I have one slide regarding that, and that's pretty interesting because we don't only interpret some sort of spatial map, but the brain interprets as well some sort of temporal map of the spiking of the neurons. Good. This is another interesting image. Here you see a brain of a mouse, and here you see the olfactory bulb, and this olfactory bulb is somehow mapped. So the blue box shows the area of the one olfactory bulb that was mapped. So here it is, this small blue box. And different olfactants, they evoke different spatial patterns of the neural activation of the bulb. So here you see the isopropyl tiglate, I guess, that smells minty to humans, and here you see the activation pattern. So here it is. That's in the olfactory bulb. So after that, glomeruli and so on, so that cells in the nose, they have already performed their job, so now we are on the second layer. So this is the spatial map that our brain will basically interpret. And here you see the ethyl tiglate. It has some fruity smell for humans. Of course, we can't ask what feels the mouse. And that activates a different pattern that you see in here. Good. So the this is a mouse, genetic modified mouse. So these cells expressed a fluorescent protein that is sensitive to the intracellular calcium levels, and the neural activity was signaled by changes in the amount of light emitted by the protein. So the colors on the maps, they represent different levels of neural activity. So if it's more hot, so like more red color, that's stronger response. If it's more bluish, that's lower response. So here you see different these different maps. What else do I have for you? Once again, a brain mouse with the olfactory bulb, olfactory tract, and olfactory cortex. Here we have a few different chemicals, two chemicals per image. The neurons responsible to the piney scent of pinene are color-coded green, and neurons responsible for the fruity scent of octanol are red. And cells responsible to both are, of course, yellow. So here you see in the upper image, here is the alpha pinene, here is the octanol, and here you see the activation pattern once again. In the lower image you see the activation pattern for hexanol, that's a cut grass smell, and the octanol that smells for us fruity or citrus. So that's once again a recording of different activation maps that the brain interprets as some particular smell. And here is the temporal coding. So the thing is that olfactory bulb and cortex, they generate oscillations when the odors are present. So there may be some sort of spike timing, there may be some rhythmic synchrony and cell-to-cell timing relationship. So the idea is that timing is an essential component of the odor identity. And there are some evidence for temporal coding of this signal, so not just some sort of spatial coding like in these maps, but we also have some sort of temporal coding. So something starts, something ends, and that old spatial temporal picture is interpreted with the brain. So we have some evidence that insects and rodents, the precise timing carries some information on the odors. We have seen that odor evoked spiked range that differ in number of spikes, their timing, their rhythmic structure, and the interesting part that disrupting some synchronous firing in bees, they lose the ability to distinguish some similar odors. So that spatial map is enough to identify something, but there is some information regarding, let's say, some similar odors that is specified with the timing. Well, that is pretty reasonable, because if they are similar, they probably activate some similar receptors, and maybe the special pattern will be more or less similar. So we need something additional. Something additional is that sort of timing that improves the fine distinction between the odors. And here you see the image that shows this sort of timing. So here we have some spiking with different neurons, and we see their temporal pattern. Here you see the pattern of the inspiration, so like breathe in, breathe out. And here you see the moment when just simple air was changed with the air that contains some chemical that has some odor, some odor 1, odor 2, odor 3. And so the spiking patterns, they may include changes of number, rate, and timing. As the mouse inspired first air and then air with odorant, the activity of 2 neurons of the olfactory bulb were recorded, and spikes from the 2 cells, so these are the blue and the red spikes, they both tended to spike twice during inspiration of air alone. So here you see that we have some inspiration, they produce just 2 spikes. Odor 1 did not change the number of spikes, but it did cause the red cell to spike much later in the respiratory cycle. So here you see how they shifted. So you still see 2 spikes. It seems like nothing changes, but you see that there is some sort of shift in timing between these 2 cells. So if we have some sort of comparator in the brain that compares timing of these cells, it will definitely know that something is going on. So here in case of odor 2, the number of spikes changed, but their timing is pretty the same, while odor 3, it changes the number of spikes. We have 4 of them instead of 2, and it changes their temporal pattern. So now they are shifted with respect to the breathing cycle. So that's what we have. So that's pretty an evidence that something is going on here. So something is going on here, and maybe this temporal information is needed for the brain to decode the exact smell of whatever chemical we are now breathing in. Good. Yes, I guess I have a few interesting points for you at the end. So let's sum up what we have. So we have our chemical senses. They are, in some sense, they are fundamental. They are pretty old from the evolutionary point of view, and they rely on some universal cellular mechanisms like second messenger systems that are used in many different cases like neurotransmission, development, and so on. There are some shared principles across different sensory systems, like broad tuning, so it's not really good if you have just individual cell that detects one stimuli. You want some population coding. there are some spatial maps in many systems, including olfactory system, despite smells have no exact like spatial information incorporated. There is what's interesting, some temporal coding. That was the last slide I have shown you, and that's pretty, let's say, interesting. Regarding the senses, before we move ahead to the last, I guess, 1 or 2 slides, I have just remembered because I forgot. Yesterday we were considering taste, and I told you that if you somehow close your nose and you try, let's say, eating some onion and some apple without inhaling the air, they should taste you much more similar than they do when you smell that. Just curious, did anyone try this at home? What were the results, if anyone has? Okay, I guess no one tried that. Okay, let it be. Good. I guess I have just few slides for you, which is basically good because I have used up almost all the time we had. These are just few funny things regarding some genes and some sort of genetic diversity that we have in the olfactory system. We have considered that regarding some taste, and that's regarding some smell. So what's interesting here? So here you see an asparagus. So here's how that looks like. Here's how it's sold in some market, for example, or whatever. And here you see how that looks in a plate. So people eat that and for a pretty long time. I guess there are some, I don't know, some ancient Roman mosaics with this sort of plant, so probably it's known to humans for a pretty long time. So what's interesting, there are some genes connected to the olfaction, notably the OR2M7 gene, and these genes, they correspond to our ability to detect certain sulfur metabolites. So it turns out that asparagus contains so-called asparagusic acid. In most people, there are certain exceptions, once again genetically determined, but most people will metabolize the thing into this list of chemicals. So here you see all of them. They all contain sulfur, and you might guess that many chemicals containing sulfur, if you see something containing sulfur, it's pretty safe to bet that it will smell badly. So I don't know, maybe worse than some sort of sulfur compounds, maybe some selenium compounds can smell. But yeah, it's most cases they smell pretty horrible. And depending on the variance of this gene, we may smell it or not. So that's why some people, so that's the mechanism in the variation in the receptor binding to the thiol containing volatiles, so the thiol group. And the thing is that some people, after eating this sort of asparagus, they smell that their pee has a very unpleasant smell when they go to the bathroom. So and interesting part that not everyone can smell that. So some people say, so, oh my god, what happened? So that's because he ate some of the asparagus, while other people, they just don't care. Another interesting genetic variation has to do with the cilantro or coriander. It has pretty interesting smell that's connected to the gene where OR6A2, There are different phenotypes, so some people smell it as soapy or pungent, some smell it as fresh and citrusy, so that depends. The OR6A2 is highly sensitive to the cilantro-aldehydes that are similar to some compounds of soap. So here you see trans-2-decenal, so this chemical on the bottom. So that's cilantro, so that's how it looks like. Once again, you can just buy it somewhere, so it's not some sort of chemical weapon. It's a regular thing that people eat. And here you see the main chemical responsible for that sort of smell. How do we know that this is ethanol? Oh, I guess I have not so many time. Okay, so here is the group, and here is all in the name. So what's interesting, that's pretty similar to the compound produced by a stink bug. So many people say that it smells like stink bugs. And what's more interesting, I went to one of the forums. It's called, I guess, Quora, and I don't remember the exact question that someone asked, but I have just collected some answers that describe how people smell the cilantro. One of the responders, he say that he's one that finds cilantro that tastes like a soap. The other one says that it's more like bugs or decay. He can't eat anything that touched the cilantro. The other responder says that he or she, I'm not sure, likes the smell of the cilantro. While the other responder says that he's okay with cilantro, but has a friend that hates it so much and tell that it smells like a cross between mince and sour vomit. So you see that some people may like it, some people don't like it very strongly. Let's say so, pretty strongly they don't like it. And the reason is the variation in the gene OR6A2. So this gene gives different sensitivity to the trans-2-decenal that is present in the cilantro, and different people, due to this reason, they describe it as pretty different smell. And I guess, yes, the last slide for today, so almost perfect timing. So here we have a few more genes. So gene OR2G3, some people, they perceive the cis-3-hexane 1O, so here it is on the right, so that's the chemical, as fresh cut grass or green leaf odor. The single nucleotide polymorphism, so maybe you have heard the term SNP or SNP, so SNPs, single nucleotide polymorphism. I guess you should have that if you're doing bioinformatics. They reduce sensitivity to this odorant, so some people consider that some pleasant fresh cut grass, some others say I don't smell anything. Interesting point, grapefruit citrus differences, like limonene enantiomers. So here you see two enantiomers, that's limonene R and limonene D. Here we have plus and minus. I hope you remember they have to do with the rotation of the polarization of the light, so plane of the polarization. Genes like OR2G3, OR1A1 and other are involved, and here you see that the plus limonene that can be found in orange and citrus, it has some citrus smell, it's found mostly in oranges, while minus limonene is found in bergamot, so maybe you have maybe you drink some tea with bergamot, so that's well, look on the internet how it looks like, maybe you will be surprised. And it has some smell like pine or tarpon. and people pretty differ in sensitivity and perceived pleasantness. So you see that these chemicals are pretty similar. They are just some sort of, well, who can tell me the exact word? What are they? Ends with mirrors. Isomers, yes, but more. Let's get more specific regarding these two. Remember our first lectures regarding the structure of the chemicals? And chiral carbon atoms. No? That's enantiomers. And this is the chiral carbon atom. And this is the chiral carbon atom. So you see they have 4 different connections. Well, one of them to the hydrogen, so it's not shown in the formula, but it's there because it should have 4 connections. So here you see that these are enantiomers, and they are some sort of reflection of each other in the mirror. So we had that, we definitely had that in the second or third lecture. And the last one thing, that's an androstenone. Oh god. I'm sorry. It has, for humans, it smells like sweetie or urinous for some people. For others, it smells like sweet or vanilla-like, and some are pretty anosmic, so they don't detect it at all. That has to do with our gene OR7D4. What's more interesting regarding these chemicals, so it's not for us, that's for pigs. So this is the first mammalian pheromone ever to be identified, and it acts on pigs. So when inhaled by a female pig that is in heat, that results in that pig assuming the mating stance. So moreover, this is now produced as an active ingredient in the boar mate. That's a commercial product made by DuPont, and it's sold for pig farmers. So if you will ever want to make some pig farm and breed pigs, so maybe you will use something like that. Good. I guess with that being said, I'm out of time and out of material. If you have any questions or statements, please fire away. Okay, I guess no questions, no statements. Good. So with that being said, have a nice evening and see you next week.